Welcome to the first NAFLD summit organized by EASL in the beautiful city of Rome, November 9th to 11th, 2017. The title of the conference is Target-Oriented Approach to Diagnosis and Pharmacotherapy of NASH, a dialogue between academia and industry. My name is Michael Trauner from the Medical University of Vienna, and I was proud to organize this conference together with my colleagues, whom I asked to introduce themselves.
My name is Klaus Hellerbrand from the University of Erlangen-Nuremberg, and I can just give it back. It was really a great, I think it was a success, and it was also fun to organize it with these three guys here.
My name is Ali Canbaya from University of Magdeburg, and it was really great topics and great meetings with a lot of discussions.
My name is Wing-Kin Sin. I'm from the Medical University of South Carolina in Charleston, in the Charleston VA, and it's been a delight and pleasure to be here with everyone.
So the way we decided to organize this conference was to put the key pathogenetic steps and processes in the center of each session, and it really covered the entire pathogenetic process, also reflecting potential therapeutic targets. And it was really exciting to see how lipotoxicity, of course, in a disease like NAFLD is in the center of interest, where we now can manipulate and target pharmacologically key pathways in lipogenesis. And it was also interesting to see that by targeting, for example, lipogenesis pharmacologically with ACC inhibitors, we actually indirectly impact on energy combustion by pumping up the mitochondrial fatty acid oxidation. And it's really the whole theme of metabolic flexibility and metabolic adaptation, which is targeted by those new therapeutic approaches. Interesting to see also how metabolism and immunity come together. There's this buzzword of immunometabolism. We have the genetic markers such as PMPL3 and TM6SF2, which also may be relevant to not only predict prognosis, but also to stratify patients and therapeutic response. So with this, maybe we make a little round of the various highlights, also of the sessions which we chaired. Klaus, what were the highlights for you?
Well, it's the other. I had the pleasure to chair the other end of the disease cascade, if you want so speaking about fibrosis and cancerogenesis. And also there we get some really exciting news. I think when you pointed out already the important role of fat in the liver. You know, we're talking about fatty liver disease. One of the things we learned is that it's actually that just the fatty liver per se alone is already a very high risk factor for the development of liver cancer. Speaking, you do not necessarily need a lot of inflammation and fibrosis. I think this has really significant impact with regards to screening of patients, this particularly, and also with risk stratification. I think that was one of the key take-home messages for me.
Ali, from your side?
Actually, we had a lot of new aspects on oxidative stress and also mitochondrial dysfunction, which are playing an important role in the progression of non-alcoholic fatty liver disease. And what we also found that people were thinking about the different cell death mechanism is not only apoptosis, which is involved, and we have other cell death modes like preoptosis, necroptosis, autophagy involved in the disease progression. And another topic which is really was highlighted was microparticles. Microparticles are really important because they have a lot of microRNAs inside which have some impact on the other cells, and these are new concepts, and maybe we will develop new therapeutic options.
Yeah, and I was chairing the inflammation and immunity section, and I think this has borne out that the role of inflammation in fatty liver disease progression from the earlier stages right up to cancer, the importance of immune subsets. Specifically, there was a lot of attention on chemokine antagonists or inhibitors to specific monocyte subsets, and also a lot of focus now on adhesion molecules involved in the recruitment of immune subsets such as the vascular adhesion protein 1 molecule. Another exciting aspects that we heard was the novel oral immune modulation for the treatment of fatty liver disease.
So we have a lot of interesting targets where we got both industry and academic perspective. We have interesting targets, but it became clear that we have to combine, that also industry has to team up. And it was interesting to see that some of the companies have different therapeutic targets within their own portfolios. Other companies have to team up with other companies. So there certainly will be also a logistic challenge in conducting these clinical trials. What was exciting for me was to see the whole revival of the field of PPARs, which with the dual agonists, where we now can target both PPAR alpha and delta, and again synergistically target various processes, inflammation, metabolism, fibrogenesis, also targeting different cells. We certainly have this exciting area of bile acid signaling that we now can target nuclear receptors for bile acids or the downstream pathways. And it's exciting for me to see how diverse this field is becoming, that we don't have just one bile acid receptor or ligand, but we have steroidal, non-steroidal ligands, which may differ in their pharmacodynamic and efficacy and also safety profiles. So this will be extremely exciting to see how that carves out in the future. And certainly there's also the link to the gut microbiota, which is a very interesting and very active field. There's no doubt that there is dysbiosis, intestinal inflammation, and changes in gut permeability driving the progression of the disease. Yet we are not very good in modifying the dysbiosis at the moment. So the study scene is extremely heterogeneous. It also was interesting to see that we have to be concerned about certain safety issues such as alcohol and lactate production by microbiota, which of course are metabolically active. And I think the major challenge here will be to conduct clinical trials that we have a refinement in terms of which probiotics or prebiotics or symbiotics really to use. The field is extremely diverse, and also have hard clinical endpoints in addition to surrogate endpoints.
Yeah, talking about this, I think in addition to therapy, we also learned a lot about diagnosis. And as you pointed out, that it's critical to combine treatments today, or that it's probably one of the potentially successful ways for the future. It may also turn out that it's necessary, or that it's at least beneficial to combine diagnostic tools. We learned about really new exciting therapeutic tools, the use of siRNAs, the use of keratins and keratin fragments in the serum, imaging technologies, how to non-invasively assess not only steatosis but also inflammation and fibrosis. So the future may actually be soon that we do not need liver biopsy any longer, or at least there are some candidates out there that are promising tools that may lead to this direction. What do you think?
But I think we should also, point out that some of the speakers state that we should be careful when we compare the mice models with the humans data. And I think this is a very important point. And another point is that the interaction between the basic science and also clinician scientists and also pharmaceutical companies, the interaction was really fruitful for all of us to maybe is as well gonna help us to develop much faster new therapeutic options.
I think, as you can see, this sets the platform for future discussions and meetings. On behalf of my co-organizers and EASL, would like to thank you all for participating in this EASL NAFLD Summit. I'd like to invite you now to log on to the EASL Liver Tree to really review some of the presentations during this conference. Thank you.